it's my pleasure to opening the afternoon session with our next keynote speaker, who is Professor Luís Carlos from the University of Aveiro in Portugal, talking today, obviously, about a very hot topic, and I can't wait to hear about how hot are living cells. So you are very well known about nanothermometry, the fundamentals and potential applications, so I guess I am, and I'm sure the audience as well. We are very excited to hear more about it.
Okay, so good afternoon. First, thank you, Eva, for the nice introduction. Okay, so I want to start with acknowledgement the organization committee, and in particular saying muito obrigado to Jorge for this fantastic meeting. Okay, so how shall I start? This is not an option, of course, because it's too late to run, but maybe we can follow some tips from all you. This is a very interesting paper written by Joss Ettinger, which she started to work during several years in this talk show, and now she is a PhD student at the University of Oxford in UK, and it's a very interesting article because she explained how the skills that she learned during the times recording television programs could be used in science. Okay, and there are several tips. Probably the best ones are that we need to tell a story. Okay, and this is what I want to do in the next 40 minutes, telling the story of some of the recent progress of intercellular luminescent thermometry is the objective of this lecture. Okay, and coming again back to Hollywood, and I quote here a sentence from Tyrion Lannister from the Game of Thrones, in which he said there is nothing in the world more powerful than a good story, and I hope that you all like the story that I have in mind. Okay, so first, before starting the story, I want to give the credit to the people in the group that works on this subject, the PhD students, which are, I think, all of them here, okay, in that row there, and also the Albano and Miguel, which are researchers, and there also, and the important role of Carlos Brites on this luminescent thermometry must be also stressed. And I will present some results that represents a collaboration with Angel Milan in Zaragoza. Okay, so this is the outline of the talk. So I will start with a brief introduction. Then I try to answer certain questions. So why intercellular thermometers are interesting, and this topic involves the measurements of local temperature gradients. So we can use some lanthanide-bearing polymeric micelles to sense these local temperature gradients. Then I will finish the lecture stressing what I think are the challenges on the topic. Okay, so why luminescent thermometers? I will pass this very briefly because there are several lectures about luminescent thermometry, okay? So I don't need to detail these things deeply, but let's stress some numbers, okay? Temperature measurements are crucial in many scientific and technological fields. It represents 80% of the world sensor market, and this is the number of that market, okay, in US dollars. This number was, in fact, a projection from 2015, and we can see in that plot the different domains. But that estimate, in fact, was achieved. You see here, this is the real number in 2020, so the estimations were really achieved, and the new projections point out an increase about 53%, due essentially to the trends in the personal computing industry, okay? Because we need smaller systems, and these systems require faster processors, and then it's necessary to monitor and controlling the heat. Okay, so we have different technologies to measure temperature, and I want to stress here that luminescent thermometry is not a remote detection method, okay? We read this several times in the literature, but this is really not true, okay? Is a remote detection method, not a contactless one. And if you look to the different technologies, we see that some of them are also contactless, like, for instance, infrared cameras. But the question is why we need nanothermometers, okay? And I think that the fast answer is that one here. So the traditional contact thermometers are not able for measurements at scales below 10 microns, okay? So if we need to work on these scales, we need to move to non-contact or remote detection methods, and among that methods, luminescence is one of the major techniques. So these limitations for small systems have, in fact, spurred the development of luminescent nanothermometers, which now lives its inflationary epoch. So we can see this in different ways. Briefly, this is a search, or this is a plot representing a data search done recently, in which we use these keywords, so temperature sensing and optical, and we can see that there are two main clusters. One is the waveguide optical thermometers, and the other one is the non-guided optical thermometers. Okay, and of course it is on this cluster that luminescent thermometry stays, and we can see that looking to the evolution in time in the last decade, so we can see that we move from the cluster, the wave-guiding optical cluster, to the non-guided optical thermometers. Okay, now if we want to have an idea about the number of papers and the number of citations, again following these keywords here that intends to cover all the field, we can see that we have an exponential evolution of the number of publications and also of the number of citations. But most important than that, if we normalize these numbers here, okay, if we normalize the citations and the number of publications using a very radical normalization, so I normalize relatively to all the papers that have in the abstract title and keywords the words luminescence or luminescent. What we found is that luminescent thermometry represents in our days around 8% of all the papers on the subject. And if you look, this number in 2020 is only 3%. So we are living clearly an inflationary epoch. Okay, so to become a consolidated subject and not a temporary fashion, I mean the community, should be able to demonstrate the importance of the technique in different domains, and I want to analyze here the specific example of intracellular thermometry. Okay, so why? Why this is important, okay? Or what are the major challenges? So I think that we can divide in two main aspects, okay? One more fundamental, okay? And the other one is the optimization of diagnosis and therapeutic processes, for instance, magnetic hyperthermia or optical hyperthermia. Okay, fundamental reasons. I learned this term, okay, thermal biology. And what it means that it's a field, like, for instance, you know, in physics, we have thermal physics, and it seems that also in biology, they start to have this thermal biology that intends to correlate temperature variation with the diverse functions of individual organisms and also try to elucidate their molecular mechanisms. So everybody knows that in our daily lives we sense a large variety of temperatures, so we can sense dry sauna, which is around 80 Celsius degree, but also we are able to sense an ice cream which has more or less minus 10 Celsius. We are also able to sense our own internal temperature, and we know that if that temperature sensing system, if it is dysfunctional, we have problems. So there is a connection linking the body temperature to the cellular temperature, okay? And that connection, for instance, in the field of spontaneous thermogenesis, so we know that the cellular activity is followed by a continuous heat generation, transmission, and dissipation, and then it is imperative to regulate and to control this heat to keep the cells operating, okay? Moreover, there are also evidences that link the response to environmental temperature not only to the body temperature, but also to the cell. Okay, so I give you two examples. One is that the response of the cells to changes on the environmental temperatures could include, for instance, the reaction of some temperature responsive proteins. And one example is the expression of heat shock proteins when, for instance, an organism is exposed to an excessive temperature between 5 and 20 Celsius degrees above the normal temperature. Another interesting example that appears recently is that this group of Chinese researchers, they use transient microscopic method to measure the heat transfer in single cells, and they found first interesting thing is that they found a dispersion of the thermal conductivity, and that dispersion is really dependent on the location in which the measurements were done. And the second interesting thing is that they report a cellular level heat regulation that responds to changes on the surrounding temperature, and this is observed in warm-blooded organisms and not in cold-blooded ones. Okay, so this is the first order of reasons. The second order are related to the methods, to the therapeutic methods. Okay, and then let's see the example of the magnetic induced hyperthermia. But to respond to this question here, I call attention that on the first day of this meeting, Teresa explaining that, okay? So for people that were not able to assist her presentation, sorry, okay? you missed a very detailed explanation, okay? So the point that I want to stress here is what are the limitations and the challenges of this method. So why this method is not applied in a more continuous way. So 2 points. First, to heat over this temperature, which is the temperature required to kill the cells, we need a high load of nanoparticles, and this is, limited by the heating power of the actual magnetic nanoparticle, okay? And I quote here one citation from this paper from 2018, in which the author said that the method has still not become part of the standard of care for cancer treatment, that there are certain challenges, and one of them is the accurate measurement of the temperature within the tumor mass, okay? And this is essential to control the therapeutic process. Okay, we, in collaboration with Angel Millan, as I stressed, we are thinking in a possible alternative to the conventional method, which involved the concept of local intracellular hyperthermia. So this concept is basically the following. So we think that we can achieve apoptosis with a small amount of heat if that heat was generated in certain locations of the cell, okay, that can trigger the cell mechanisms leading to apoptosis. For that, we need to create a temperature gradient near the nanometer, okay, which could be, for instance, a magnetic particle that hit the surroundings due to the alternating magnetic field. And then if we have a thermometer at the surface of this particle, we can sense these local temperature gradients. Okay, the subject has attracted the attention of a broad audience, including science journalists, and we can see here, for instance, one paper on the scientist, and a second one more recently on the ACS journal. Okay, so let's move and let's discuss one of the things that interest most the community, which is the possibility of having local temperature gradients inside the cells. So I will present two examples. The first one is that one here. So this is a paper from 2011 in which the authors use quantum dots. They use the spectral shift of the emission of the quantum dots, and we can see here that they try to follow the increment of the temperature following calcium 2 plus stress, and also following a cold shock to the cells. So we have in these graphs the dispersion of the increment of temperature before and after the calcium addition, and we can see here that before calcium addition there is no increment of temperature, and then suddenly we have an increment reaching after a plateau, and this is of course not happens in fixed cells because there is that process that does not occur. What I'd like to stress here is that there is an inhomogeneous intracellular temperature progression, okay? And this also occurs after a cold shock experiment, so they lowered it suddenly, the temperature of the cell from 37 to 15, and what they have is a significant broadening of the temperature distribution in live cells. We can see here the temperature distribution in fixed cells is much, much narrow. Okay, second example of these intracellular temperature gradients, the authors here use a nanodiamond. They increase the temperature exciting the system with near-infrared irradiation, and they detect the increment of temperature using the optically detected magnetic resonance. And what the results show is that there is an intracellular temperature increase which is inhomogeneous, okay? And they estimate that increment of the temperature inside endosomes, because the particles, the nanodiamonds, are incorporated in the endosomes. So that increment of temperature was not less than 30 degrees. Okay, then these are two examples of the experimental results. Then this paper was published in 2040, and these are theoretical people, and basically what they said in this paper is that it is not possible to have these increments of 10 or even 2 degrees, okay? And then, of course, this motivate strong reactions, that there are replies to this paper and replies to the replies of this paper. And I can say that we try to send a reply to a reply, and the editor of the Nature Method answered to me saying that no way, I want to close that discussion, sorry, but I don't want to publish nothing more about this discussion. Okay, let me show here or stress to you part of a review paper written by Uchiyama Group, in which they try to argue why the theoretical paper is not true, okay? But basically, that the argument is that in cells, we have a very complex medium, and in that very complex medium, we cannot apply a simple expression discussing the heat dissipation based on the Fourier law. But I don't want to take any one of the positions, okay? My message here is that this is a completely open question, and I hope that I can give some more details about that. Okay, but I think that this cartoon illustrates well the situation. So by one hand, some people believe that they are able to measure these intracellular gradients, and by the other hand, there are some people that say, no, there is no intracellular temperature gradients. The problem is that you are measuring wrong. Your method is not appropriate for this complex medium. Okay, so what is our contribution to this discussion? So in brief, as I mentioned, this is a work in collaboration with Angel Milan and also Rafael Pinol. And we use polymeric micelles. The point here is that the micelles have an hydrophilic and an hydrophobic part, okay? And is on this hydrophobic part that we coordinate there. We coordinate lanthanide-based complexes, okay? Complexes based on europium and samarium, and then we form this type of micelles, okay, in which the complex, the light-emitting centers, the centers that we will use for sensing temperature are very well protected for the interaction with the surroundings. Okay, so these are some structural data showing that we are, in fact, working with micelles of an average size around 20 nanometers. Okay, and these are the excitation and emission spectra. I call attention that we will use emission from europium that lies in a region that is superimposed to one filter. I will explain later this better. And the samarium emission was superimposed to a spectral region which is coincident to a second filter. So this is the temperature dependence of the emission, okay, one complex and another one. We were able to explain this temperature dependence. Again, I will not detail the mechanisms of luminescent thermometry. Andreas do this in the first day, and again, if you are not there, sorry, you missed a very, very interesting lecture. So what I want to stress here is that we use a ratiometric method to sense temperature, okay, because we have europium emission and samarium emission, and the basis of the mechanism is what we call energy transfer driven thermometers, and we were able to explain this temperature dependence of the europium and samarium level in both BTFA and DNPD micelles, okay? And this work was done by Albano Neto, which is here, okay, which can explain that variations in the intensity due to these different mechanisms from the ligand levels to the lanthanide ions, and we have not only energy transfer from the ligand levels to the levels of the lanthanides, but also the back transfer, which is also temperature dependent. Okay, so the main point is that we were able to explain this temperature dependence, and when we move to the intracellular experiments, we realized that the BTFA complexes are not good, because you see the intensity of the emission decreases strongly, okay? But on the DNPD system, the intensity is more or less similar, so the two spectra are very, very similar, and you can see here that this is the calibration curve in water, and the calibration curve in the culture medium is very, very similar. I will come back to this point later. So with this, and using this setup in which we use a light-emitting diode to irradiate the sample, and then we collect the emission of the sample, and we divide that emission here in two channels, one the europium channel and the second one the samarium channel, and we use that two filters that I mentioned previously to separate the emissions. And then using a flux diagram that analyze the two images, the images of the europium channel and the one of samarium, we construct a ratiometric image, and then we apply the calibration curve, and we have the thermal image. So for the fixed cells incubated with the micelles, here are the results. So you see the results are independent of the concentration of the particles, and we have a very narrow temperature distribution as we expected, okay? But when we move to the living cells, first we can see here that the temperature distribution is much, much broader, which is in agreement with some of the results that I shown before, and we have some brighter points in the cells, and then if we sense the temperature there, okay, which is these red dots, we realize that the temperature is higher, okay? The distribution is more or less with the same full width at half maximum than the blue dots, but the temperature is higher, okay? We were able to localize the micelles in the nucleolus of the cell, and then our propose is that these red points represent the temperature of the nucleolus, which are a little bit higher, around 5 degrees higher of the temperature of the rest of the cell. And we can also see in this picture that if we increase the culture medium temperature, okay, both temperatures, I mean the temperatures of the cytoplasm and the temperature of the nucleolus, they follow the medium culture temperature. Okay, then last part of the talk. Remember this picture from Andreas' talk, okay? I should say here that Andreas is the responsible for that title, okay? This is his merit, because I think that this is a fantastic title, and so when we discuss this in our Nano-TB Tech webinars, we have in mind the luminescent thermometry, okay? But what I want to stress here is, can we trust in intracellular temperature measurements through luminescent thermometry? The answer to that question is, I don't know. So first, we have a very complex medium. The thermometric parameter must be independent of any input other than the temperature, okay? And we should verify that. And finally, how to get the calibration curve in the intercellular medium. Because I cannot put a reference thermometer there. So there are a lot of questions or challenges. I acknowledge Danny Yak because in our early morning runs, we discuss a lot of things, and one thing that we discuss is these problems, and he sent to me a big table in which he and the member of his group record some data from the literature, and you can see that there are papers in which they don't have a calibration curve in fixed cells. So, and then they use a calibration curve. In some cases, it's not clear even the procedure to get the final temperature. I will give you 2 examples only. This paper, it's very interesting because the authors argue that the mitochondria work at the temperatures close to 50 degrees. So, and there are a review paper that discuss this problem. Second example, this is a paper in which this group, which is by the way, that they work in Portugal, they use magnetic hyperthermia experiments, and they conclude that they have temperature heterogeneities around 30 Celsius, and when they apply the alternate magnetic field, they can reach temperatures above 70 degrees. What I think, one thing that could be the problematic is the calibration curve, as I mentioned before. So this is the conventional approach. So people have experimental points, then they fit the experimental points with an empirical equation or with the Boltzmann equation, and then what it means that when they move from one medium to the other, they need to repeat the calibration. But if we are in an intracellular environment, this could be very, very difficult, okay? And then what typically people do is assume a unique calibration relation. And this is clearly a bottleneck. So for that, maybe the concept of primary luminescent thermometers can help, okay? But up to now, nobody used this concept to solve or to try to help to solve better the problem. Okay, examples of people that confirm in dead cells and alive cells and in suspension that the calibration curve is more or less the same, okay? In this case, these people use a core shell structure, and again, the light-emitting centers are well protected for the surroundings. But we have also the opposite. So for instance, in this case, this is the calibration curve in suspension, and this is the calibration curve in vivo. And you see despite the units here, this is the logarithmic of the intensity ratio, the values are quite different, okay? So, or in this case here, in which they use quantum dots, and you can see that the temperature response in a live cell or in solution are quite different. So, This means that this is a problem or a challenge that needs to be addressed. Finally, the other pitfall is that we need to verify the independence of the thermometric parameter with, for instance, again pH or again ionic strength and the refractive index, because in a living cell, the refractive index could change a lot between one point or the other, okay? In this paper, the authors do this, or did this, but I should say that in the great majority of the papers, they don't say nothing about this independence. Okay, so take home message. First point. This is okay. I am a strong support of the luminescent thermometry. So there are really interesting devices with a lot of potential applications in different areas, ranging from nanomedicine, but also microelectronics and nanofluidics. However, as Andreas stressed Monday, we need to start to define standard guidelines. Okay, so not only on the experimental procedures, but also on the way that we treat the data and making this clear. To become a consolidated subject and not a temporary fed, we need to demonstrate how important the method could be as a tool for different subjects, for instance, for thermal biology or to study heat transfer at the nanoscale. And I believe that intracellular nanothermometers are a nice example to applicate this tool, okay, and to show how the problems could be very complicated and or very complex, and how we need to work together to try to solve these problems, not only on the setup of the measurements, but also on the treatment of the data. Okay, so let me acknowledge the founding agencies and, of course, all of you for the kind attention. Thank you.
Thank you very much for this wonderful presentation, and we have time for some questions. Yes, here. No, for some. I will be strict on time, but...
My name is Mario Diaz. I'm an orologist, a molecular orologist.
Oh.
I'm an oceanophysicist. But I still find this very exciting, and I find your talk was really brilliant and very well explained, and very clear for even not experts in the area.
Thank you.
During more than 30 years cultivating cells in vitro, I have never managed to have cells alive below 35 degrees, not above 38 or 39 degrees. But I know these are culture conditions, and this is very experimental. So I would have a suggestion to all of you working on thermometers, intracellular thermometers, to work on closer to real, to physiological ranges in order to have these results more, with more impact in the field. Working 40 degrees above or below the physiological temperatures are, playing with artifacts. So it's just a suggestion to all of you. The other thing is that I kind of belong to the group of people having concerns about the variation, the heterogeneity in the temperature. But on the sense, this is something that the organism, especially in homeotherms, is very well controlled. But on the other side, as a molecular biologist, I completely agree with part of the messages you have mentioned today, that there are differences in in the intracellular temperature because there are mechanisms to regulate mechanisms like producing heat for the whole organism that have the origin in intracellular compartments. This is the case of mitochondria. Mitochondria is a central power unit in the cell. And the cell has the ability to block and to uncouple the sort of reactions in the electronic chain to produce more or less heat, or in fact, more or less ATP. So if you measure close to the mitochondria, then you will probably have changes in the degree of uncoupling, and this is something that in thermodynamic terms means that you increase the enthalpy of these exothermic reactions by reducing the entropy. And this means an increase in heat. And that is one way that animals, even cold animals, have to regulate the whole body temperature, for instance, in the brain, in some kind of fishes.
Okay.
So I would just suggest, or I would ask, if you have managed to develop a methodology to measure intracellular, but in close proximity to mitochondria. And not in the nucleus. The nucleus, for instance, the nucleoli that you have used is a very packed structure of chromatin, and so it's got no activity at all.
Yeah, okay, to answer directly your question, I think that this is probably the evolution of the intracellular thermometers, in the sense that they need to have a way to target specific points in the cell. As far as I know, this system that sense the temperature of the mitochondria are molecules, are small molecules that are fluorescents that goes directly to the mitochondria. But in this case, there are some concerns about if the thermometric, if the parameter that is used to measure temperature, there are doubts if it depends from other inputs. You know, so this is the, as far as I know, the actual stage of the field.
Thank you very much for this nice overview and your work. From a specific case, the micelles made with samarium and europium, I understood that there was no fluorine in that case. So you don't need the, let's say, the nanoparticles. Is that correct, or am I wrong? You just need the ions, the samarium and europium.
Yeah. But the point is that we need to have the centers, the ions, very well protected. Otherwise, that emission will be strongly dependent on the conditions of the medium, and we can also have a strong quenching. So this is why we use that complexes, because first we can excite on the ligands, and then the energy transfer to the ion is very efficient, and the cross absorption on the ligands are very, very high.
And now it comes my question, and probably you gave me already the reply. If they are, in this case, encapsulated at the surface of magnetic nanoparticles, so very close to the surface, what can be the problem? The fact that they quench because of the magnetic absorption, so that would be not a system to propose, because there are ways that you can, let's say, bring these ions at the surface of the magnetic particles. But would be the quenching a problem?
No, yeah, this is that paper that I mentioned, which is now in revision, but we are fighting about the referees, and I'm not sure if we will be able to publish the paper, at least in ACS Nano, but let's see, because one of the referees doesn't believe at all in our results. But in this case, we have the magnetic particle, the core, okay, and then the the complexes are located at the surface. And we found, due to the fantastic job done by Rafa, we found a system in which the magnetic field does not alter the luminescence. Okay, so we are able to have, these are images of in living cells, and we are able to see the emission of the europium and of the samarium ion.
Thanks, and thanks for the interesting talk. It's perhaps a kind of philosophical question, but I have that discussion sometimes with people from the magnetic hyperthermia community, and that is, shouldn't we measure energy or absorbed heat and not temperature? So shouldn't we kind of try to sum up all the energy in the cell and what we have put in there, and that way try to determine the heat distribution or something like that?
Yeah,
thank you.
Thank you for the question. What I can say to you is that this part was the design of the system for measuring the emission during the application of the field was designed in Zaragoza for a group of people, and one of them is Abelardo. Abelardo is one of the pioneers of the induction magnetic induction for cook, So, and one thing that I remember that we discussed several times is that it is really important that the heat is generated by the magnetic nanoparticles and not by the Joule effect of the setup. And this is, of course, an important thing to take into account.
Do you have a clue why people don't accept or do not use this pretty simple and very useful concept of primary thermometers?
I think it's our fault. I mean, our fault, my fault and the fault of the people in the group, because we are not, good enough to show to the community that this is simple and it can be very useful. So I was really very, very happy, and I was not the opportunity to congratulate Marta, but I will do this now, because Marta was probably the first one outside Aveiro that used the concept. So it was really a moment of happiness this morning. But maybe it is, it's difficult to people have in mind a certain methodology, okay? And then they follow that methodology, and it's difficult in certain cases to broken this.
All right, so obviously your talk triggered a lot of questions and need for discussions. I think we can continue this during the coffee break. So thank you very much again for your wonderful presentation.
Thank you.